Hello friends, welcome back to my YouTube channel Doctor's Corner. In this today's video, we are going to discuss about action potential of the nerve fiber. So in the previous videos, we have discussed about the resting membrane potential in a nerve. Now we are going to discuss about the action potential of the nerve or of the neuron. So in this particular video, neuron or nerve, we are going to discuss about the definition of the action potential, the phases of the action potential, and its ionic basis of this phases of action potential, and also how the action potential propagate or the conduction or propagation of the action potential. Okay, propagation or conduction of the action potential. So with these objectives, we are going to discuss the today's topic, that is the action potential of the nerve. Okay, now first coming to the definition of the action potential, what exactly the definition of the action potential means. Okay, so to understand this definition, I'll just draw a rough diagram of a neuron. So consider this as a neuronal cell. Okay, as a neuronal cell, and the fluid which is present outside, it is called as ECF, that is extracellular fluid, and the fluid which is present inside, it is called as intracellular fluid. As you know, there are so many ions present inside the cell as well as outside the cell. So inside the cell, if you do the sum of all the ions and you do the sum of the ions, cations and anions on the outside the cell, so at the resting condition, when the cell is not stimulated, that is when the neuron is not stimulated, at the resting condition, the resting membrane potential is minus 70 millivolt. Okay, so resting membrane potential of a neuron is minus 70 millivolt. Why it is minus 70, why it is negative and all, there are so many factors we have discussed under the video of resting membrane potential. So please go back to the playlist of general physiology and nerve muscle physiology, and please watch that video of resting membrane potential to have a clear concept of that. Then come back to this particular topic, action potential. Okay, so how we will record this resting membrane potential? So generally it is recorded by a instrument. Basically it is a voltmeter known as cathode ray oscilloscope. Okay, so if we keep the two electrodes on the surface of the cell membrane, so there will be generally no deflection, that is no electrical activity deflection or potential change between these two electrodes is seen. Okay, but on the other hand, if we insert one of the electrode inside the cell, okay, inside the cell, and record the membrane potential at the resting condition, then the interior of the cell is negative with respect to the exterior, that is called as resting membrane potential. That is the membrane potential change at the resting condition is called as resting membrane potential. So for the neuron it is minus 70 millivolt, for the skeletal muscles and all it is minus 90 millivolt. For different tissues or different cells, the resting membrane potential varies. Okay, now when this particular excitable tissues, okay, in excitable tissue means which can be excited or stimulated, in which the signal can be propagated, for example neuron and muscles like skeletal muscles, cardiac muscles and smooth muscles or some few glands, these have a property of getting stimulated or excited. Okay, that is why they are known as excitable tissue. So when an excitable tissue is stimulated, for example this neuron is stimulated by an electrical current, okay, with the help of a battery, if you stimulate the cell membrane, then there is opening and closure of certain ion channels which are present on the membrane, and it will causes the movement of the ions either from outside to inside or from inside to outside, and it causes some electrical changes. So these electrical changes or the sequence of changes which occur briefly, that is the brief sequence of events which occurs rapidly on the membrane potential of an excitable tissue when the cell is stimulated, that it is called as action potential. Okay, so what is the definition of the action potential? It is the rapid changes which occurs on the membrane of an excitable tissue when the cell is stimulated, it is known as action potential. Okay, so whenever the cell is stimulated, some electrical changes, that is from minus 70 it will change to plus 35, and again it will come back to minus 70. So that rapid changes in the membrane potential when the cell is stimulated, it is called as action potential. Okay, now we are going to discuss about the phases of action potential. So why this action potential is important, why we need to study so many things, is because the conduction or the transmission of the impulses in the excitable tissue, for example, this we are discussing this neuron. So how this neuron message, that is how the message from the neuron is transmitted from the one part to the another. For example, I am standing here. If suppose there is some touch sensation or pain sensation or thermal sensation, it is hot or cold, this sensation from this my hand will go back to my central nervous system, that is ultimately the brain. In detail we'll discuss about that pain pathway, touch pathway, temperature pathway and all later. But how this signal will go through the nerves, right? Through the nerves it will go to the spinal cord, and again through the spinal cord obviously it will go up, that is the sensory receptors are stimulated here, and ultimately it will go up to the brain. So how this signal goes from one neuron to the another neuron and reaches the brain, and the signal goes, or the nerve transmission takes place with the help of this action potential. So what is action potential? It is an electrical phenomena which causes, when the cell is stimulated, there is a rapid change in the membrane potential. Now coming to the phases of the action potential. Okay, so I'll just draw a graph to understand this phases of the action potential. So on x-axis, that is the time duration. So I'll just draw this as the minus 70 millivolt, here minus 55 millivolt, it will be plus 0 and up to plus 35. Okay, so at the resting condition, if we record with the help of the cathode rays oscilloscope, the voltage difference across this cell membrane, that is the RMP, resting membrane potential, is minus 70 millivolt. Now if we stimulate this neuron, okay, suppose there is a peripheral neuron, if there is a touch sensation, suppose if the magnitude or the power or the force of the touch sensation is very minimal, okay, so if there is a touch sensation, what generally happens is there is opening of certain sodium channels. Okay, there is opening of the sodium channels, and the sodium from outside, that is the ECF, will enter the ICF. Okay, then the interior of the cell will tend to become positive, that is from minus 70 it will become minus 60, minus 65, minus 50, like that. Okay, now if the strength of the stimulus is very less, then it will the current will again come back to the normal. Why the current will again come back to the normal? Because of the presence of very important structure, one of the structure which will maintain the RMP, though it is not the main reason, but it is there, known as sodium potassium pump. We have discussed in detail in the previous video of the resting membrane potential. Okay, so sodium potassium pump again bring back the membrane potential to minus 70 millivolt. But when the strength of the stimulus is adequate, that is, if can have a response, it can have a response. For example, the threshold stimulus is minus 55 millivolt, that is, if the strength of the stimulus is enough, which will reaches the threshold stimulus, then there is the opening of the voltage gated sodium channels. Please give your concentration here, voltage gated sodium channels. Okay, so when the stimulus is adequate, that is known as the threshold stimulus. Okay, if the stimulus is inadequate, it is known as sub-threshold stimulus, then the current is not enough, and this potential what is generated is not the action potential, it is called as graded potential or local potential. We'll discuss about the difference between the graded and the local potential in the some other video. Right now we are discussing only the action potential, so won't discuss about this graded potential in this particular video. Okay, now the action potential, when the neuron is stimulated by a threshold stimulus, so what will happen is this voltage gated sodium channels are opened, and the sodiums from outside, that is the extracellular fluid, will come inside the cell. Okay, will come or enter inside the cell. This entry of the sodium, that is the positive charge from outside to inside, causes the positivity inside this cell. Okay, now this positivity inside the cell, it is called as depolarization phase, and slowly this positivity will reaches from minus 70 up to the plus 35 millivolt. So it will try to go to the equilibrium potential of this sodium, that is nearly plus 60 millivolt, but around plus 35 millivolt it will stop because of the closure of the sodium channels. Now you should have a very clear idea how this sodium channels opens and closes. Okay, so consider this as a cell membrane. Okay, consider this as a cell membrane. Now what I am drawing this as a bilayered cell membrane. Okay, on this cell membrane, consider this as the sodium channels. Okay, consider this as the sodium channels in this blue color. These are the sodium channels through which sodium ions enter from ECF to ICF. Why sodium channels will enter from ECF to ICF? Because concentration of the sodium is more in the ECF as compared to the ICF, that is the intracellular fluid. Now in the resting condition, there are gates which are present on the sodium channels. This gate which is present on the ECF, it is known as activation gate. Okay, activation gate generally, and this one more gate or which is present in the ICF, it is known as inactivation gate. Okay, inactivation gate. So at the resting condition means when the cell membrane or the membrane potential is minus 70 millivolt. Resting condition means when the cell membrane is minus 70 millivolt. This is the structure of the gate, that is activation gate is closed. That's why the sodium ions are not coming inside the cell and the membrane polarity is maintained at minus 70 millivolt, and it is called as in a polarized state. Now what happens when this cell is stimulated? So this sodium channels gets activated means what exactly will happen is, so activated means what happens to the gate is this activation gate will open, and this inactivation gate will slowly try to, it's like a hinge, activation and inactivation gate. So when it tries to open slowly the inactivation, when it is like this at the resting condition, so when the activation gate opens, inactivation gate slowly try to close it. Okay, so during this phase when the activation gate opens and inactivation gate is still open, okay, then what happens is sodium ions from outside, it will enter the ICF, that is from the ECF, sodium ions will enter into the ICF. Now this is called as activated phase. So this activated phase generally it occurs from this minus 70 millivolt to plus 35 millivolt. Okay, generally. So when the sodium channels are open, generally not from minus 70, that is from minus 55, when it reaches the threshold potential, then this will occur. Okay, so this, when this opens slowly, so from this threshold potential, that is from minus 70 or minus 55 more specifically, when it reaches a threshold stimulus, then only this voltage gated sodium channels are opened, and from that it will now goes to the equilibrium potential of the sodium, which is around plus 60 millivolt. But by the time when it reaches the plus 35 millivolt, that is when the action potential which is recorded by the cathode rays oscilloscope is around plus 35 millivolt, what happens is this sodium channels, that is this inactivation gate closes. Inactivation gate closes means it will be like this, that is this inner side inactivation gate will be inactivated state, that is this inactivated gate closes and activated gate will be still open. Okay, now this condition usually occurs at the phase, that is when the membrane potential is at around plus 35 millivolt. Again it will come back to the resting membrane potential, so that is minus 70 millivolts. So till that the condition will be of the sodium channels will be in the inactivated condition. Okay, it will be in the inactivated state. So sodium channels which are present on the cell membrane, okay, so this is the cell membrane, and these are the sodium channels. So at the resting condition, the activation gate is closed and inactivation gate is open. Okay, it is open, so even then the sodium ions can't enter. But when the cell is stimulated by the threshold stimulus, then this voltage gated sodium channels activation gate opens and this sodium will enter inside the cell. So after reaching some time, slowly this inactivation gate is closed and the sodium ions can't enter it and sodium channels are closed. Okay, so this phase, it is called as depolarization phase. So depolarization phase is mainly due to the opening of the voltage gated sodium channels. Now during this time, when the sodium channels are closed, then there is one more channels which are present on the cell membrane, which are known as voltage gated potassium channels. Okay, voltage gated potassium channels. These potassium channels also, which are present on the cell membrane, generally they will be in a closed state. There will be in a closed state. Okay, so on a cell membrane, this voltage gated potassium channels are also present. This green color are the voltage gated potassium channel. So at the resting condition, it will be like this, that is in a closed state. So no movement of the ions takes place. Now when their depolarization has reached up to plus 35 millivolt, that is the inside of the cell has become plus 35 millivolt, then what happens is this voltage gated potassium channels now open. So voltage gated potassium channels now open because of the opening of this gate, okay, at plus 35 millivolt. Okay, at plus 35 millivolt, this will get opened up to when it reaches till minus 70 millivolt. Okay, so it will be open, and the potassium ions from inside, it will go to the outside. Why from inside to outside? Why not from outside to inside? Is because the concentration of the potassium is more inside the cell. So the potassium ions will move outside the cell when it is stimulated, and because of that the charge, positive charge, is moving outside the cell. So the cell will now tend to become negative, and this negativity of the cell causes the membrane potential to reach to the resting membrane potential, and this phase it is called as repolarization phase. So what is the reason of the repolarization? It is the closure of the sodium channels. So during this repolarization phase, sodium channels are in the inactivated state, that is inactivation of the sodium channels and opening of the potassium channels. So potassium channels open and sodium channel close, and the membrane potential becomes negative. So it is a very slowly opening potassium channels, and it will even surpass this resting membrane potential. Again it will come minus 80, minus 90, and further finally it will come back to the resting membrane potential because of the sodium potassium pump and the other factors. Okay, now this it is called as hyperpolarization phase. Okay, hyperpolarization phase. Again it will come back to the resting membrane potential. So if at all one more time electrical stimulus is given, then again there is a wave of depolarization, then wave of repolarization. Okay, so we have studied now the phases of action potential. So two important phases of action potential is depolarization and repolarization. Depolarization, it is due to the opening of the voltage gated sodium channels, and repolarization, it is due to opening of the potassium channels and simultaneously closure of the sodium channels. Okay, there is again one more phase of hyperpolarization. Again finally the membrane potential will come back to the resting membrane potential. Okay, this is how this membrane potential changes takes place, and this membrane potential changes, it is called as action potential. Now how this action potential is transmitted in a myelinated and unmyelinated nerve, that is the propagation of the action potential. Okay, so again I am drawing a rough consider this as a neuron, okay, neuronal cell. Okay, so on this patch of the membrane, there are sodium channels as well as the potassium channels will be there. Okay, so there will be different patches will be there on this. Consider this only one segment or the one part of the cell membrane. Okay, so suppose if this particular part of the cell membrane is stimulated, by an adequate stimulus or an electrical stimulus, now what happens is, so this one, okay, part 1 or segment 1, then what happens is first sodium will enter and again potassium will go. Sodium entry causes the depolarization and repolarization. You will get a typical action potential curve like electrical changes, right? Now this depolarization in this particular segment, because of the entry of the sodium ions during the depolarization phase, the sodium ions will now open the neighboring sodium ions, okay, in a positive feedback manner. Okay, so this again further in the next segment will open and the sodium ions will enter in this segment 2. So again from this it will again stimulate the opening of this voltage gated sodium channels, and finally the repolarization will occur, depolarization, repolarization. So in this way the membrane action potential is transmitted in an unmyelinated neuron. Now in a myelinated neuron, so in a myelinated neuron means generally Schwann cells, SHWAN, okay, Schwann cells are the one which will help in the myelination of the peripheral nerves or the neurons. So basically what they do is it acts as an insulator, and they will wrap. So this blue color thing is basically a Schwann cell which will wrap itself around this neuron, okay, which will wrap itself around the neuron, and this layers of the Schwann cells which are there, they are called as myelinated neurons, okay. So this Schwann cells, so in the peripheral neurons or in the peripheral nervous system, the Schwann cells will causes the myelination, whereas in the neurons of the central nervous system, oligodendrocytes will causes the myelination, okay. So neurons in the central nervous system will be myelinated by the oligodendrocytes, and here by the Schwann cells, okay. So they will form this one, like and basically the cell membranes are the lipid structures, sphingomyelins and other will be there, and they will be having they will be an electrically insulating materials, that is they will be lipid in nature. Now suppose this myelinated neuron, this part is stimulated, that is action potential generated, sodium and potassium ions are stimulated. Now inside will become negative, but this can't be propagated because of this myelin sheath. So instead of this and it will block the propagation. Now from interior the sodium channels will go and they will stimulate this or opening of the voltage gated channels here. Again this will enter here and they will stimulate the voltage gated sodium channels here, and the action potential is propagated in this manner, that is they will jump from one part or the one node to the another node, and these junctions they are called as node of Ranvier, okay, they are called as node of Ranvier. And this type of propagation while jumping, okay, from one node of Ranvier to the another node of Ranvier, it is called as saltatory conduction, okay, saltatory conduction. So if we analyze the propagation of action potential in an unmyelinated neuron and myelinated neuron, so the impulse, that is the speed of conduction or propagation of the electric current or the action potential is more in the myelinated neuron, because it will jump from one neuronal cell, this one from one node of Ranvier to the another node of Ranvier, like type A fibers, which are myelinated fibers in which the conduction or speed is very fast, okay, and whereas the unmyelinated neuron, the conduction will be slow, okay, like fast pain and slow pain. We will discuss about that pain pathways when we discuss about the pain pathways. So right now just remember, in the myelinated neuron the conduction will be fast because the action potential will be jumping from one node to another phone. This is all the cell membrane, okay. Now there is one demyelinating disorder known as Guillain-Barré syndrome. We will discuss about that in some clinical videos. So in that what happens is, this myelin sheaths are this one degeneration, and the neurons will become demyelinated, and that conduction of the tissue is not there. Even in this there is a one layer of the myelin, that is the Schwann cell will wrap in this unmyelinated neuron, but in the myelinated neuron Schwann cells form the multiple layers, okay. We'll discuss that details in the video of the Guillain-Barré syndrome. So right now just to summarize in this particular video what we have discussed: the definition of the action potential. What is action potential? It is the rapid changes in the membrane potential following the stimulus of the particular excitable cell or a tissue, okay. That is, it is a brief changes of electrical events that occurs when the cell is excited or stimulated. The phases is depolarization, repolarization and hyperpolarization. Again come to the resting membrane potential. Ionic basis also we have discussed. Then how it is propagated in the unmyelinated and non-myelinated neuron, okay, or unmyelinated neuron and myelinated neuron, okay. So if you like our video, please subscribe our channel and share it with your friends. Thank you.